Now, the meeting is being live streamed, so welcome people out there in TV land. We meet the second and fourth Thursdays of the month from late September through early May, excepting holidays and a winter break. And we hit 5 years in the spring, and there's a QR code so you can donate. There's also a link in the chat. And tonight's session is Upending Assumptions About Life on Earth with Saul Gruner. In December, we'll have How Has Your Science Changed in the Last 5 Years with a panel of previous guests on science in the virtual pub. And our first session in January will be the session rescheduled from October 22nd or 23rd, I believe it was, with Suzanne Macy talking about why we need to conserve biodiversity and how improving our teaching will help. And with that, I'm going to stop sharing and hand it off to Saul. Take it away, Saul.
Okay. Let me start sharing. Okay, can you see the shared screen?
We don't see it yet. There we go. Now it's coming.
Okay. So I want to thank Don and PRI for allowing me to talk to you today. So please grab a drink, virtual or otherwise, settle back, and let's jump right in.
And I will pour my drink as you get started.
So in 1962, Thomas Kuhn, a science historian, wrote a very influential book titled The Structure of Scientific Revolutions. In it, he posited that science proceeds by paradigm shifts. A paradigm in this context is some foundational understanding of some branch of science. And a paradigm shift is when that understanding becomes outdated and it's replaced by new understanding, a new paradigm. So as an example, a century ago, geologists were wondering how mountains formed. And the prevailing paradigm was that the early Earth started out as a ball of molten rock. It cooled, it developed a rigid crust, it continued to cool, and it shrank. And as it shrank, the crust wrinkled like the skin of a drying apple, and that's how mountains were formed. And then in 1915, a German scientist by the name of Alfred Wegener proposed that you could understand a lot of geologic puzzles if you simply assume that the continents drifted over the surface of the Earth. We call that continental drift. Now, Wegener was ridiculed. People said, that's crazy. What force could possibly move a continent? But over the next 50 years, evidence accumulated, and by the mid-1960s, is unambiguous that, in fact, the continents do move over the surface of the Earth. And a new paradigm, what we now call plate tectonics, had settled in. Well, what I'd like to do today is try to convince you that we're in the midst of a paradigm shift about where much of life on Earth exists, what's required to sustain it, and how life evolved. So let me walk you through the history of it into the implications in the second half of this talk. If you're going to talk about the evolution of life, of course, you have to start with Charles Darwin. Darwin thought very deeply about evolution, and he couldn't help but ask himself the question, well, if life evolved, what did it evolve from? He had speculations, but no data, and he was far too careful a scientist to put speculations in publications. But to his friend J.D. Hooker, in a letter he wrote, but if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes. In other words, Darwin was pointing out that if life evolved from an unliving Earth, it had to evolve from the primordial materials that were available at that time. But chemistry in Darwin's time, late 1870s, was really quite primitive. You really couldn't put together a lot of the details. In fact, if you think about it, even by the beginning of the 20th century, there were still great debates about whether atoms existed. But by the time you get to the 1920s, after World War II, chemistry had advanced quite a bit. Sufficiently so that this man, a Russian scientist by the name of Alexander Oparin, wrote a book called The Origin of Life, where he detailed a lot of the chemical steps of taking smaller molecules and making larger molecules. About the same time, a British polymath by the name of J.B.S. Holvey, a guy with a kind of remarkable mustache, was positing similar ideas, apparently unaware of Oparin's work. But these were, in both Oparin and Haldane's cases, theoretical, chemical theoretical speculations. What was needed was experiments. That came along in 1953 when Stanley Miller, a graduate student at the University of Chicago, was working on an idea posited by his PhD mentor, Harold Urey. He's the guy who won the Nobel Prize for discovering deuterium. He did a simple experiment. He took a glass globe and he filled it with gases that were thought to represent the atmosphere of the Earth a long time ago: water vapor, hydrogen, ammonia, and methane. And in this glass globe, he had inserted 2 electrodes, which were connected to a very high voltage source, so a spark was constantly jumping between the electrodes. The idea being that the energy of the electrical discharge would break apart these molecules into their constituent atoms, which would then randomly recombine in other ways. He put a condensing jacket on the bottom of this apparatus, which condensed the water vapor into liquid water, ran into a flask where it was heated and turned to steam, and that drove circulation around this apparatus. And what Miller noticed is that the water, originally crystal clear, in just a few days of running this apparatus turned murky brown. And when he chemically analyzed the constituents, he found that indeed the water had been filled with all kinds of organic molecules, including amino acids, which we know are the basis for proteins. So this led to the current paradigm, the one that I'm going to try to argue is being overthrown, which we might call the recipe for life. You start with a reducing atmosphere, reducing the sense of oxidation reduction reactions. You expose that atmosphere to energy that can break apart molecules, heat from volcanoes, ultraviolet from the sun, lightning. And then you form a host of molecules, organic molecules, which can be thought of as monomers. And they would accumulate in the waters as a form of primeval soup. That term was coined by Oparin. And then, with a gigantic leap of faith, somehow these molecules come together and form the larger polymers of life: the lipids, the nucleic acids, and the proteins. Now, what I want to draw your attention to is that there's an assumption built into this paradigm. That assumption is that all this is happening at the interface between the atmosphere and the waters. That was built in, if you think about it, into Darwin's conception as well as Oparin and Haldane's conception. But it is an assumption. What happens if that assumption is wrong? Let's explore that. Well, first of all, why do people assume it? They assumed it because they thought they understood what the biosphere was. It was the oceans, lakes, and land surface. And the reason they thought that is because everyone believed that life depended ultimately on photosynthesis. Energy from the sun is used by photosynthetic organisms to take carbon dioxide and water and make organic molecules. That's the basis of the food chain. People believed at the time that the deep Earth's crust, the rocks that are deep under our feet or underneath the bottom of the ocean, were sterile. They thought that because it's dark down there, light doesn't penetrate rock, there's no photosynthesis. So they thought there's no form of food. And furthermore, as you go down into the Earth's crust, the pressure and temperatures start rising very rapidly. And that was thought to be very inimical to life. That set of assumptions was blasted apart in 1977 by the discovery of deep sea hydrothermal vents, proverbial black smokers. So these occur at the spreading zones of oceans where magma comes up from the mantle and forms basalt, basically new ocean crust, and spreads from that point. This forms cracks in the rock. The waters at the bottom of the sea go down to these cracks. They encounter the very hot rocks. Of course, this is kilometers down, so the pressure is very high. Hot, high-pressure water is very corrosive, and it leaches all kinds of minerals and gases from the hot rock. This then rises to the floor of the ocean where this water comes out, often as hot as 400 degrees centigrade, and encounters the much cooler, near freezing temperatures, the ocean bottom, and the much more alkaline water that's there. In doing so, a lot of the stuff that's dissolved inside the water comes out in the form of precipitates. The smoke that you see in the black smokers is, of course, not smoke. What it really is particles, mostly metal sulfides, coming out of the plume. This astonished the people who discovered it. But what really blew their minds were these living communities that they found around the hydrothermal vents, creatures and forms that no one had ever seen before. And the natural question they asked themselves was, wait a minute, there's no source of photosynthesis down there. Light only penetrates a few hundred meters into ocean water. So what are they eating? Didn't take too long to analyze the waters and realize that the base of the food chain were microbes that were consuming the gases and other things that are coming out of these hydrothermal vents. In other words, they had discovered a community which had no solar dependence. It was completely independent from the sun. Furthermore, over the ensuing years, people realized that these things exist all across the planet. Okay, there are many, many hydrothermal vents, actually of different types as well. Some people said, well, where are these microbes and this life coming from? It was initially assumed that these were surface evolved organisms that happen to take advantage of a new food source. Life is very opportunistic. And so they adapted to this. But some people said, well, how do you know that they aren't organisms that came from the other direction, perhaps from the rock up? How do we know that? Well, there are assumptions built into that. If you look into the literature over the previous 2 centuries, there are roughly 4 dozen papers where people had examined microbes that they found in deep wells, oil wells, for example, or deep mines, gold mines, and posited that, in fact, these microorganisms were growing inside that deep rock. These papers were uniformly dismissed. They were dismissed because the authors could not counter an objection, which is whenever you go into deep rock from the surface, you always start at the surface, of course, you have the potential contamination of surface organisms. So how did these authors know that what they were seeing were not contaminants that came in from the surface? That was the basic objection. If you really wanted to answer the question of is there life growing in deep rock, you have to do broad, sustained, and systematic studies, and that takes a great deal of money. It takes a great deal of money because mining and drilling is very, very expensive. So what you really needed was something which drove the need to look into the rock. And it turns out by the mid-1970s, that need had materialized. That was trying to get rid of nuclear waste. So by the mid-1970s, nuclear waste were accumulating in countries across the globe, from the Cold War and from reactors that were being built to produce electricity. And the problem was then, what do you do with it? It's intensely radioactive. You have to sequester it from contact for thousands or tens of thousands of years. This was also the peak time of the environmental movement. The environmental movement really had flourished throughout the 70s, and people become very sensitized to the obvious thing to do with waste is you bury it very deeply. Deep rock where you suppose it could stay for millions of years. People said, well, if you're going to do that, how do you know what's down there? Perhaps you should look and check. So I believe largely to assuage public opinion. In 1986, the US Department of Energy funded a guy by the name of Frank Wobber to create something that they call the Subsurface Science Program, or the SSP. And the idea was to fund this with enough resources to really do systematic tool development in order to study if there is deep life in rock. And the waste problem was an international problem. Other programs were going on in other places, other countries around the earth. The challenge that Wobber's team and others faced was try to answer the question, do microbiota live inside the deep crust? And of course, by now it was clear that you shouldn't assume things. You actually have to sample and analyze and come up with watertight arguments. But in order to sample the deep crust, you have to tunnel or drill down there. Now, we've all seen pictures of people coming out of mines. We've seen pictures of people working on oil well rigs. They're covered with dirt, schmutz from head to toe. I mean, it's a really filthy business. So if you want to bring up samples, you have to convince people that they're uncontaminated. Well, how do you do that? Well, one of the first things that Wobber's team was develop a series of coring tools. So imagine that you drill down, you rent an oil well rig, you drill down a mile or two, you pull the drill out, you lower this coring tool, which they had invented, which would hollow out or core out a piece of rock that was perhaps a few inches across and 6 or 10 inches long. The quarry tool would then pull this rock into a sterile chamber inside itself. This would be brought to the surface, where Waber's team had erected a sterile laboratory, basically a gigantic glove box. They would insert the tool inside the glove box, extrude the core. They had tools inside that glove box which would strip away the outside of that cylinder of rock, leaving the core in such a way that they thought it would be uncontaminated. But of course, you have to convince people that it really is uncontaminated. How did they do that? They developed tracers. Now, there are a lot of tracers. Perhaps the most important one in this context are perfluorocarbons. These are low viscosity liquids that have two important attributes. The first one is that they don't exist anywhere naturally on the surface of the Earth. They're completely synthetic. And the second one is that you could spectroscopically determine the presence of these perfluorocarbons at a volume of 1 part in 10 to the 15. It's really sensitive. So you might imagine the way this went. They would drill down, bring up the drill bit, pour some of this perfluorocarbon liquid down in the bottom of the well, lower the quarry tool, take a rock core, bring it up, insert it inside their glove box, extrude it, test the outside for perfluorocarbon. Of course, it's completely contaminated with that stuff. That's a control. And then strip away the outside of the rock and test again. And if they don't see any perfluorocarbon, then you have a pretty persuasive argument that you've not contaminated the sample. But this immediately brought up a second problem, which is, how do you find a few microbes in a kilogram of rock? Again, it's a problem of 1 part in 10 to the 15 by volume or so. The normal way that you would deal with microbes is you would grow them up in culture until you had a significant quantity of them, and you start to analyze them. But nobody knew how to grow these organisms if there were organisms there in the first place. But fortunately, by this time, the mid-1980s, second technology had come to the rescue, which is the polymerase chain reaction. Now, we have all heard of the PCR reaction, especially after COVID. It uses an enzyme that was extracted from a microbe that was found in Yellowstone National Park and in the thermal pools. And by cycling a solution that might have DNA with it, with this enzyme and other appropriate chemicals dissolved in it, you can exponentially multiply up any DNA that might be present. Even a few molecules can, in a relatively short period of time, a day or two, be multiplied up to quite significant quantities. So people started taking the sterile core, breaking it up, washing it through with sterile liquid, and then running that through a PCR machine to see if they saw DNA. And they saw lots of DNA. In fact, by looking at the sequence of the DNA, you could get some hint about the organisms. And what they found, in many cases, these were organisms no one had ever seen before. Okay, so it really does appear that there's life in deep rock. People started doing more and more experiments of this type, to the point where in 1992, Tommy Gold, an astrophysicist at Cornell University, in fact, he has his office, or had his office, he's now deceased, in the building next to the one I'm sitting in at the moment. He felt compelled to write an article that appeared in the Proceedings of the National Academy of Sciences of the United States. And Gold said, there are strong indications that microbial life is widespread at depth in the crust of the Earth, just as such life has been identified in numerous ocean vents. This life is not dependent on solar energy and photosynthesis for its primary energy supply, and is essentially independent of the surface circumstances. Its energy supply comes from chemical sources due to fluids that migrate upward from deeper levels in the Earth. In mass and volume, it may be comparable with all surface life. Now, that last sentence is a real kick in the head. And in truth, Gold was bluffing. He really had no evidence to support that last sentence. But it was intriguing and it provoked. And people started doing more and more experiments. More and more wells were drilled, more and more deep mines were analyzed. Let me now fast forward 25 years to 2017, when a second paper appeared in the Proceedings of the National Academy. Now, print small, let me read this for you. 25 years ago this month, Thomas Gold published a seminal manuscript suggesting the presence of a deep hot biosphere in the Earth's crust. Since this publication, a considerable amount of attention has been given to the study of deep biospheres, their role in geochemical cycles, and their potential to inform on the origin of life and its potential outside of Earth. Overwhelming evidence now supports the presence of a deep biosphere ubiquitously distributed on Earth in both terrestrial and marine settings. As Gold suggested, it has now become increasingly evident to better understand the subsurface is critical to further understanding four things: Earth, life, the evolution of life, and the potential for life elsewhere. And I think we all have to admit those are in fact some very big, deep questions. First question one has to address is, what's the energy source? What's feeding it? Well, we know that the Earth is far from thermodynamic equilibrium. It has reactive minerals, it has primordial heat left over from its formation, and energy is constantly being added from the decay of radioactive elements, uranium, thorium, potassium-40, that are distributed throughout the Earth's body. In fact, if you think about it, we have solar energy coming in. That's a high-density source of energy. It's on average on the order of a kilowatt per square meter surface area. But there's also energy coming up from the Earth. It's 1/10,000th of that magnitude. It tends to be about 1/10 of a watt per square meter, but it's constant. And it's been there basically ever since the Earth formed.
Randy Keel.
In 1992, going back to Waber's team, they did manage to extract and culture a microbe. It was anaerobic, doesn't like oxygen. That's good because there's no oxygen in a deep well. It's thermophilic. That's good because it's hot down there. They dubbed it Bacillus infernus. No one had ever seen this organism before. Infernus from inferno. And they finally figured out how to culture it, what they could feed it to make it grow. They presented results about this at 1993 at an International Symposium of Subsurface Microbiology in England. See, by 1993, there had already been enough reports going on that scientists felt that they had to get together on a regular basis to discuss these results. So when Waber's team presented, other people supported this, was that it was eating hydrogen. Now, where is the hydrogen coming from? It was proposed that the hydrogen came from the radiolysis of water. Radioactive elements inside the rocks would send out energetic rays, alpha, beta, gamma rays. These would encounter water molecules and split them apart, evolving oxygen and hydrogen. So in a funny way, these microbes they proposed were eating radioactivity, which I find really ironic, given the fact that you might recall that the motivation for Waber study in the first place was to think about what happens if you put radioactive material down there. We now know that there are additional sources of hydrogen. In addition to the radiolysis of water, certain minerals, when they're fractured, will evolve hydrogen. And importantly, olivines, which make up a very large fraction of the mantle of the Earth, when brought up by tectonic processes, often convert to serpentines, another mineral, and in the process they evolve hydrogen. So there's been a constant source of food, and this constant source has been going on for billions of years. So the next question that naturally comes to mind is, okay, there's biomass down there. How much? By the time you get into the second decade of the 21st century, enough studies have been done, enough wells analyzed, enough deep mines analyzed, that enough papers published that people could start doing meta-analysis of what people were finding in various places across the Earth and try to figure out how much biomass is down there. The results in some sense are startling, at least I find them startling. The estimates range from a few percent to a majority of the Earth's biomass. Now, the notion that a few percent of the Earth's total biomass might be in deep rock, I think is amazing. The notion that it might be a majority of the biomass, I mean, is absolutely mind-blowing. Let me give you some papers and show you some of the things that people have been saying. So this is a paper that came out in 2018, and it shows in very schematic form how different biomes gave rise to different forms of life. So the axis here is time. This is the fraction of the total biomass of the Earth. Today, we believe that most of the biomass of the Earth, at least I'll go into this in just a moment, exists in plants. In fact, most of that is wood. But, you know, plants and forests have been on the Earth's surface only for less than 500 million years. The Earth is 4.5 billion years old. Prior to that, the subsurface really dominated, people believe, okay, the total biomass of the planet. This is not a decrease in the biomass, for example, underneath the deep continental rocks. Rather, the fraction of the Earth's total has decreased relative to an increase because of plants. Now that's schematic. Let me give you some real numbers. There's another paper came out also in 2018 in PNAS, where they quantify, based on various papers that have been presented, what these biomass distributions were. And they point out that on average, people were seeing 470 gigatons of carbon in terrestrial sources. Again, that's mostly wood. A gigaton is just a way of quantifying the amount of biomass. Imagine extracting all the carbon from the biomass and weighing it. Okay, a gigaton of carbon is roughly a cubic kilometer of solid carbon. The marine component is actually quite small, 6 gigatons of carbon. The deep surface, subsurface, these two areas, it's estimated to be 70 gigatons of carbon. That's a lot. That's on the order of 15%. Now, earlier I said from a few percent to plausibly a majority of the Earth's biomass. What justifies that statement? You've got to realize that these are estimates based on averages of what people saw, but they have very large error bars, especially for this component, because you really have only drilled, even today, a few hundred places of the Earth's surface. Okay, so it's a quantity of numbers. There simply isn't a lot of data of which you could tighten down error bars. The same people who wrote this paper listed the error bars. Here they are. Plants, that's mostly wood, 450 gigatons of carbon, with an uncertainty of a factor of 1.2. We know, because we could see, where this biomass is. We could measure the forests, we could measure the organic matter inside the soils. But look at the bacteria, most of which are in that deep subsurface. The uncertainty is a factor of 10. Maybe it's 7. Maybe it's 700 gigatons of carbon. The same thing is true for archaea, microbes that until the 1970s were thought to be bacteria, but we now know that they're a completely separate branch of life. So the uncertainties will contract out as more and more holes are drilled, more and more mines are examined. I've been noticing over the years that these numbers have been creeping up. Who knows where they'll settle? But sufficient to say that at present, the best estimates are 15%, but that number may very easily change. Once you realize there's a lot of biomass down there, then think about it just a little bit. You find there are a whole bunch of grand challenge questions that come to mind. I'm going to walk through these in just a moment, but I first have to give you a caveat. Answers to these questions that I'm about to pose will be years in the making. This is really the work of the coming generation. So if you're a young scientist listening in, take heed. There's lots of work to be done. The data and papers are coming out at an increasingly rapid rate. Most of the ones I've shown you, most of the ones I will show you, were done in the last 10 years. So the best I can do at this point is to give you a snapshot in time of where I think we are based on the literature that I've gone through, the literature that's out there. Some of the things I'll tell you will certainly be wrong. Things that I can't think of will have popped up, okay? That's the way science is. We're at the cutting edge of it. First question, did life start in hell? That is to say, in the deep, hot, high-pressure environments that you have in the deep Earth's crust. Very interesting paper came out in 2016, Nature Microbiology. By 2016, there are enough whole genome sequences of the three branches of life, the bacteria, the eukaryotes, and the archaea, all of animals, and most large plants, of course, eukaryotes, that people could ask the following question. Let's analyze these whole genome sequences and see what genes are common amongst them. And if we could find the common genes, then perhaps we could learn something about the environment in which their common ancestor lived. Okay. In the literature, the common ancestor is called LUCA, it stands for Last Universal Common Ancestor. Why do we believe there was a common ancestor? Well, there are too many things about these three branches that are the same. They all use the same DNA. They use the same genetic code. They use the same amino acids. The amino acids have the same handedness. I mean, there's a long list of chemical similarities, which could be happenstance, but seems extremely unlikely. The simplest explanation is if they all evolved from last universal common ancestor. They analyzed millions of genes. from databases that have accumulated over the years. And they found a few hundred. Then they asked, what do these genes code for? What kind of proteins do they code for? And what do these proteins do? And it turns out that they were the kinds of proteins that you see in these organisms living around hydrothermal vents. Now, an objection was raised because these organisms can trade DNA. It's called horizontal gene transfer. But that also can be addressed. They did so in another paper. Their conclusions are as follows. It's from the paper. Did the origin of genetics hinge upon hydrothermal chemical conditions that gave rise to the first biochemical pathways that in turn gave rise to the first cells? Genes that trace to LUCA, ancient biochemical pathways, and aqueous reactions of carbon dioxide with iron and water all seem to converge in a similar set of simple exergonic chemical reactions as those that occur spontaneously in hydrothermal vents. From the standpoint of genes, physiology, laboratory chemistry, and geochemistry, it's beginning to look like LUCA was rooted in rocks. We already know from work that's been done in recent years that the Miller approach will never work. Okay. There are a variety of reasons why it won't work, but the simplest is that as you start making larger and larger molecules for monomers, then these larger molecules have more atoms, and therefore they have more interatomic bonds. And therefore, they have more targets to be broken apart by the energetic sources that broke apart these molecules. So the process becomes ultimately self-limiting. You have to think of another way of putting smaller molecules to larger ones that exists in some environment that you have on Earth. Many people at this point are converging on the notion that the most promising environment that you could come up with are these hydrothermal vents. They meet the criteria that you really want in order to have this process occur. It moves material down a free energy gradient. And in doing so, it compartmentalizes that material in a series of compartments which naturally occur in hydrothermal vents. So people are beginning to believe, and I think at this point the weight of evidence is shifting in this direction, that hydrothermal vents are a good environment for meeting the constraints of that leap of faith that Miller had, how you get from smaller molecules to larger molecules. Still much work to be done, but progress is being made. We know that hydrothermal vents have been around for a long time. So here's a paper came out in 2017 in Nature. Although it's not known when or where life on Earth began, some of the earliest habitable environments may have been submarine hydrothermal vents. Here we describe putative fossilized microorganisms that are at least 3.8 billion, and possibly 4.3 billion years old in ferruginous iron-containing sedimentary rocks, interpreted as seafloor hydrothermal vent-related precipitates from this place I could never pronounce in Quebec, Canada. This place is actually where we find the oldest rocks on Earth. Now, people who do this kind of work are constantly arguing with one another whether squiggles that they see preserved in fine-grained rock are signatures of microbes. And the arguments go back and forth. But what is not being argued is the presence of hydrothermal vents dating way back to early Earth. So did life start in hell, deep down in high pressure, high temperature environments? Well, we don't know, okay, but it's looking more and more like that's plausible. Furthermore, hydrothermal vents look like a possible, most plausible biome or region that people have come up with that meets the constraints of making larger molecules out of small molecules. Second question, when did life start? And what does it take to sterilize the Earth? Now, that second part of that question, was it take to sterilize the Earth, may strike you as strange. Let me show you why I bring it up. First of all, let's look at the Earth's chronology. It's key to this. So here's one drawn from Wikipedia. It's very typical. The first 500 million years of the 4.5 billion years of the Earth's existence, called the Hadean. The next 1.5 billion years is the Archean. The next 2 billion years are the Proterozoic. And finally, the last 500 million years is the Phanerozoic. This is where basically anything that you could hold in your hand that's large enough in a single organism and see it came out in that last 10 billion years. Now, we know that the Earth formed about 4.5 billion years ago. We know that because there's great reason to believe that meteorites and asteroids, things that fall from the sky that we can hold in our hands, formed at the same time as the solar system in a relatively short period of time. And we could date the radioactive decay of atoms that are inside those meteorites, and they converge upon this date. Within a very short time after the proto-Earth formed, it's believed that this proto-Earth collided with a Mars-sized planetoid. It's been given the name Theia. And those two completely obliterated one another and then recoalesced in the Earth-Moon system. The evidence for this comes from the Apollo astronauts. Their job when they landed on the moon, aside from getting there and back, was to bring back rocks. And the rocks that they brought back looked remarkably like Earth rocks. And this started to answer a series of puzzles that astronomers had been wondering about literally for centuries. The Earth-Moon system is unique in the solar system. There is no other planet in the solar system which has a moon that's anywhere near as large a fraction of the parent body as the Earth and the moon. They formed in a very different way, it's thought, than the moons that orbit other planets. By 4.4 roughly billion years ago, we believe the Earth was covered by oceans. Why? Well, there are no rocks that date back that far, but there are mineral grains that do. The rocks have been recycled through tectonic processes. There really is very little in the way of rock, nothing that we found that goes back 4.4 billion years. But there are mineral grains, in particular zircons, zirconium silicate crystals, often sub-millimeter in size, that do date back that far. How do we know that? Well, it turns out that zircons have a very desirable characteristic. They're loved by geochronologists for this characteristic, which is the zircon has a lattice that is very susceptible to incorporating uranium. And so a zircon crystal actually has enough uranium in it, and by using accelerated mass spectrometry, you could do this in crystals that are really only a bit larger than a red blood cell. You can analyze the two isotopes of lead that result from these decay processes and accurately date that zircon. And we do have zircons that date back to this period of time. Furthermore, when crystals grow, they tend to incorporate bits of their environment in the form of inclusions. And some of those really old zircons have minerals in them that we know are only formed in marine environments. So this is the evidence that leads us to believe that the Earth was covered by water very early on. No one knows where the water came from. It turns out that that's a big puzzle. When did the earliest fossils, did life form on Earth? Well, the earliest fossils that are relatively unambiguous are in this area of time, 3.5 to 4.4 billion years ago. These fossils, the ones that are not disputed, really look very much like microbes that exist on Earth today. Furthermore, they're stromatolites, these accretions of sand, bacteria, and photosynthetic organisms that currently exist. We see stromatolites growing in Australia, and we find evidence of stromatolites that are in these old rocks. That means that by this period of time, there already was life on the planet that had been fully formed. That's to say, it had the same kind of features that we see in life today, single-celled organisms. Probably DNA, probably a lot of the same biochemical processes. That means life had to form earlier. So the community is sort of converging on the fact that life probably formed towards the end of the first 500 million years of the Earth's existence. This brings up a problem. Problem is that if life formed that early, how did it survive something called the late heavy bombardment? Late heavy bombardment, if you haven't heard of that, let me walk you through it. Again, let's go back to the moon. If you look up at the moon when it's full on a clear night, you see the face of the man in the moon, as it's called. What you're really seeing are these very large craters. Now, 3 of the Apollo missions landed in these craters to bring back rocks, and they tend to date to this period of 4.1 to 3.9 billion years ago. It appears that during that period of time, there was an unsettling in the solar system. It's now believed that it had to do with the migration of one of the outer gas planets. We don't know which one. Maybe in Saturn, maybe in Jupiter, maybe in Uranus, which perturbed the gravitational equilibrium that had settled in. And it caused large rocks that were in the outer part of the solar system to change their orbit, sending many of them towards the inner solar system where they bombarded the inner planets. We also see in radar maps of Mercury similar craters. The moon and Mercury preserve these because it doesn't have plate tectonics, which would destroy them. It doesn't have weather, which would erode them. If we now ask about the kinds of impacts that cause these, they're really quite catastrophic. We've all heard of the Chicxulub event. This is the impact of a rock that's estimated to be 5 to 10 miles across that hit what is now the Yucatan Peninsula of Mexico and wiped out the dinosaurs about 66 million years ago. Let me give you a brief description of what happened on that day. This rock hit the Earth. It's just a tiny speck compared to the size of the Earth, but it carries a lot of energy, equivalent to some trillions of tons of TNT. In doing so, it blasted a crater tens of kilometers deep. It vaporized hundreds, if not thousands, of cubic kilometers of rock. These went up into the stratosphere, and this cloud of hot rock spread around the Earth. As this hot rock came raining down, it raised the temperature at the surface of the Earth where it fell to hundreds of degrees. If you were an animal that could not find shelter in a cave or underground, you were fried alive. If you were a plant or a forest, you burst into flame. It was a global conflagration. After the heat ended, didn't last very long, everything burned, you put a tremendous amount of soot into the atmosphere. Along with that pulverized rock, the Earth went dark. There are various estimates for how long it went dark. Days, months, some people say years. Photosynthesis collapsed. Organisms that managed to not be fried found they no longer had photosynthetic food. It was a mass extinction. It's estimated that 85 to 90% of the species on Earth disappeared. This kind of collapse, this kind of cataclysm is small in many ways compared to the ones that would have happened during the late heavy bombardment. If the moon was bombarded, so was the Earth. The Earth is 50 times as large a gravitational steric object. Of course, we don't see the craters because of erosion and plate tectonics have gotten rid of the evidence. But by looking at the size and number of craters on the moon, you could estimate pretty well what had happened to the Earth. Here's one of those estimates. The late heavy bombardment of the Earth would have had more than 22,000 impact craters, diameters about 20 kilometers across. The Chicxulub event was 200 to 300 kilometers across. 40 impact basins with diameters of 1,000 kilometers. I mean, that's enormous. And several diameters of 5,000 kilometers. It's hard to conceive of how energetic an impact like that is. This would boil the oceans. So how would life have survived this? The initial assumption was it didn't, but not so fast. If you're buried in deep rock, unless you happen to be right underneath where the impactor hits, you're pretty well insulated. And at this point, the science of being able to understand the impact craters numerically, things that have been validated by large thermonuclear bombs, is pretty advanced. So you could estimate with this distribution of cratering over a several 100 million year period, what would happen in terms of making the Earth sterile. Here's a report where that's done by several groups now. So let me just read the bottom line. Our analysis shows that there's no plausible situation in which the habitable zone was fully sterilized on Earth, at least since the termination of the primary accretion of the planets and the postulated impact origin of the moon. So, when did life start? Well, it's beginning to look like it started within the first 500 million years of the Earth's existence. What does it take to sterilize the Earth? A lot. And chances are it hasn't been sterilized since life first formed on the planet, at least that period of time. How long is a microorganism viable in these deep environments? How long does it take for a cell to reproduce? Now you realize these cells are living inside a very different environment in very different circumstances than we're used to. Furthermore, their energy sources are constant, but they're low flux. So they're living very slowly. Here is a report that came out 2017. It was done by a group, Japan and Caltech. The Japanese are superb high pressure scientists. They have the best deep sea drilling ship, scientific drilling ship in the world. What they did in this case is they went off the coast of Japan, where the Asian plate is being dragged down by the subduction of the Pacific plate inside a deep ocean trench. I believe it was the Japan Trench. They went down through some kilometers of water and then through 2 kilometers of continental shelf, and they encountered coal. That's not surprising. It's continental shelf rock. Coal was 20 million years old, and when they analyzed it, it contained living microbes. At this point, they asked a very clever question. People had analyzed lots of microbes from that were brought up from ocean floor in the middle of the oceans. They said, and they don't look like the kind of microbes you have in continental rock in general. Do the microbes that are in this coal look like microbes that you would have in forests, or they look like the microbes that we find in the center of the ocean? And the answer was, genetically, they look like forest organisms. In other words, 20 million years ago, there were trees growing in some swamp. Those trees fell into the swamp where the waters had low oxygen. Eventually, those trees were covered by sediment, compacted, dragged down, converted into coal as these things, as the continental shelf margin was being dragged down into the earth. And all along, these organisms were evolving and adapting and continuing to live and reproduce. They tried to analyze how long it takes for these cells to multiply. How long does it take for a single cell to double? You could do that, for example, by feeding it radioactive tracer foods and seeing how it incorporates it. All they were able to say at the time is that the incubation times were estimated to be from months to hundreds of years. Work since then has found that in some cases, these organisms reproduce once every few decades. That doesn't exclude longer reproduction times for other organisms. It's just really hard to do this work. It's very hard to convince graduate students to do a PhD thesis on an organism that does something once every 100 or 200 years. Okay? It's not very exciting work, but it's very important work. They're living very slowly, basically metabolizing and catabolizing their molecules just to avoid the kind of thermal degradation that occurs naturally. And then every once in a while, they decide to split. The arguments within the community is what the maximum time for a single microbe to multiply is 100 years or 1,000 years or 10,000 years. We really don't know the answer. It's a very hard series of experiments to do. So life is very robust. Well, it's actually really, really robust. Here's another experiment that was done. These people went into halite, rock salt. Rock salt, as it's found on the surface of the earth in many, many places, is basically some arm of an ancient sea that became landlocked, dried down to complete solid salt, then got overlaid with sediments that are watertight. Otherwise, the salt wouldn't have lasted through time. The salt crystals, like all crystals, tend to include a little bit of their environment. Remember the zircons and the minerals. Well, when you make halite, you tend to include little bubbles of brine. You see it in little bubbles that are in all halite formations. And what these people did is they went into a Permian salt bed in the Permian Basin of Texas. It's the same region where a lot of the oil comes from. And they drilled into one of these little brine inclusions. There's the drill hole. They extracted what looked to be bacterial spores that had been locked inside this brine inclusion since the salt formed a 250 million years ago. And they reported on the isolation and growth of a previously unrecognized spore-forming bacteria, which they gave the very exciting name 2-9-3, from a brine inclusion inside this salt. So stop and think about this. These bacteria were encapsulated, they went into a spore state, and they hung around for a 250 million years until these scientists came along and released them. Now, whenever I talk about this, somebody brings up Jurassic Park. Have no fear. Halite beds are everywhere. In fact, as I sit in my office at Cornell University, I'm 1,000 feet above an extensive rock salt bed that goes all the way from here to Ohio. These things are eroding all the time. Whenever you put rock salt On roads, of course, you're putting a lot of this stuff and letting it disperse. So these bacteria are being released all the time. This experiment, had they looked a little bit deeper in the literature, actually was done 40 years earlier or so by a German by the name of Heinz Dombrowski, who analyzed the Permian salt bed in Germany. And he claimed that he was able to grow bacteria from brine inclusions as well. But at that time, he didn't have the genetic tools we now have to be able to date the DNA and show that it really was that old. So how long is a microorganism viable? Well, we don't know the answer to that question. In spore form, it's certainly viable for a good fraction of a billion years, and we don't know what the upper limit is. There's a report in the literature of 780 million years that has yet to be verified, but it's a long time. How long does it take for an organism to reproduce under these conditions? We already know that it's tens to hundreds of years and maybe thousands. We really don't know the answer to that. Furthermore, if you think about these spores, they are able to survive extreme conditions, because the next question you would ask is, what are the pressure and temperature limits of life? How hot can it get, and how high can the pressure get before life becomes impossible? Again, we really don't know the answer to these questions. We already encounter and find in various places deep life organisms that are very happily getting along at 120 to 130 degrees centigrade. I remind you that at high pressure, water is not boiling, okay? But they've adapted to be able to have their biochemistry go on at these very hot temperature limits. For pressure, even fewer experiments have been done. You could take microbes in the laboratory, subject them to pressures which are many thousands of atmospheres, corresponding to depths of many miles below the surface of the Earth, bring them up, and they're still, in many cases, viable. We don't know how long they would be viable, because people haven't done experiments where you pressurize them to high pressures and leave them around for many years. Again, these are difficult experiments to do. But it's already clear that although we don't know the ultimate pressure and temperature limits of life, they're certainly not below the boiling point of water, and certainly not below the kinds of pressures you encounter, at least for the top few kilometers of the Earth's crust. In fact, in the spore form, it's almost certain that as continental margins are being dragged down into the mantle by tectonic turnover, that there are spores. It may be a one-way trip to hell, but there are spores that likely are still viable. They may never see the surface again, but it's something to think about. Here's another question. It's one of my favorites, and it's complete speculation. But as you'll see, it's informed speculation. Does non-DNA life coexist on Earth? All life that we know is based on DNA. These organisms I've been talking about, in many cases, we've only known about them for a relatively small period of time. How do we know about them? Well, we know about them primarily through the polymerase chain reaction. That's an enzymatic reaction, which, like most enzymatic reactions, is very specific as to a substrate, DNA. If you had a polymerase chain reaction and you had some genetic material which is not DNA, then that genetic material would not be multiplied up and you would not see it. Chemists have been very clever in coming up with alternative polymers, nucleic acid analogs, okay, which potentially could convey genetic information. So, is there non-DNA life coexisting on Earth? Well, if we're honest with ourselves, how would we know it? At this point, no one has really looked. We can now ask the question and try to figure out ways to answer that question. But really, we cannot answer this question in the positive or the negative. Give you a context for this, let me just tell you about a certain bacteria, the genus Pelagobacter. Some of you may have heard of it, especially if you're a microbiologist. If you haven't heard of it, don't worry, very few people have. This organism was first detected by DNA or nucleic acids in seawater in 1990. It wasn't until 2002 that they managed to culture and grow a particular species of Pelagobacter. But we do now know the following. Pelagobacter exists in abundance in all seas. When I say abundance, I mean it's estimated that half of the summertime bacterial load that you find in seawater is probably Pelagibacter. Furthermore, it's very heavily involved in the cycling of organic carbon. It's very important inside the carbon cycle of the Earth. So if you've swum in an ocean, if you've gone swimming in and licked your lips, you know how I've consumed thousands of these things. Until very recently, we didn't know they exist, even though we were surrounded by them all the time. So why would we know about life that might exist in these deep environments when we haven't really looked at it? My point is simply that we should do some thinking about how to come up with experiments to answer this question. Wouldn't it be great if all these years we've been looking for aliens up there, if in fact the aliens were coexisting with us all the time down there as obligate piezophiles? They exist only at high pressure and high temperature. Just interesting things to think about. Again, I emphasize speculation. There's no evidence for or against that. How does deep life differ from surface life? Well, we know it differs in many fundamental ways. It's hard to study because if you want to study microbes, you really have to grow them up to sizable quantity, watch their life cycles, see their biochemistry in action, and so on. DNA gives you some hints, but it doesn't tell you the whole story. Now, people for years have been going down sampling deep sea water and trying to grow bacteria, and they've had a very hard time about it. Karen Rogers at the Rensselaer Polytech Institute outside of Albany has come up with a very simple set of experiments. They're actually complicated in detail, but they're simple in principle. She's come up with very innovative plumbing. So she lowers steel bottles, for example, down into the bottom of ocean trenches, opens them up, and sucks in some high pressure water, then closes them up. Because they're rigid bottles, they can be brought to the surface without the pressure being relaxed. And in her laboratory, she's created plumbing where she can attach these bottles to that plumbing, force the liquid into various nutrient media, and see if they'll grow, never having experienced a drop in pressure. The hypothesis being that if these are obligate piezophiles, things that require high pressure for their molecules to work well, that they may have died, and that's why people hadn't been successful before. And lo and behold, she's finding all kinds of bacterial organisms that people had not seen before. So there's a lot of work to be done here, but it's very clear that a lot of these organisms really need the pressure in order to be able to exist. What are the effects of high pressure on biomolecules? The reason these organisms are obligate piezophiles, they need high pressure, is because biomolecules are strongly affected by pressure. Now, you may have noticed I've been giving you a history. I haven't told you at all about what I do. This is actually one of the things that I study, the effects of pressure on biomolecules. And I'm not going to tell you about it other than the fact that there are many effects. I'm not going to tell you about it because this is a short talk and I've run out of time. But if you're interested, in 2022 to 23, I was a fellow at the Hamburg, Germany Institute for Advanced Study. And one of my goals in going there was to develop a series of lectures about high pressure structural biology. What you've just heard is a lecture intended for the general public on why we should study high pressure biology. By now, I hope that you're convinced that the reason we should study is because it represents a lot of life on Earth. And if we really want to understand life on Earth and the questions that I posed so far, we really want to understand these biochemical processes. The two other lectures, which were technical lectures, were on high pressure structural biology, primarily using X-rays as a way of determining how these molecules change their structure when you pressurize them. Because what we've learned is that the key to biological function for large molecules is to understand their structure. If you're interested and you want to explore this talk is being recorded. Don could lead you to where it's found on YouTube. You could always pull up this slide. If you go to this website, or alternatively, you put in that QR code, it'll lead you to the extended lecture, number 1, questioning assumptions about life on Earth. And this is on the website of DAISY, which is the National High Energy Physics and Hard X-ray Laboratory of Germany. On the upper right-hand corner, you'll find a search box if you were to go to this site. If you put in my name, Gruner, Gruner, you'll pull up all 3 lectures. Finally, science is a social activity. Okay. And I've been very fortunate over the years to work with really fantastic students, postdocs, and colleagues, both at Princeton University, which is where I was for the first half of my career, and at Cornell University, the second half of my career. And I'd like to acknowledge and list them here. And with that, I want to thank you for your time and attention. I'll be happy to try to answer any questions that you might have.
That was great. Thanks, Buzz. And we've already got a question in the chat from George Adams, which I will read. I would assume the environment at thousands of feet below the surface does not provide voids in which colonies of these microbes might populate. Thus, they are spread quite thinly in the crust. Would this lead to absence of any detectable fossils of the organisms, despite being ubiquitous and ancient enough to ride the parting tectonic plates to the subduction zone, where some should have scraped off to wind up in coastal accretionary wedges?
There actually are voids inside deep rock. The voids are able to withstand the pressure because they're filled with water, and water is very stiff. It's not only in deep rock, by the way. People have been surprised when they started drilling into the ice in Antarctica. As you may know, there's an experiment where they built a neutrino observatory by drilling 1 kilometer down into the ice in Antarctica. And people were surprised to find that even in that deep ice, which has been locked in deep form for many, many years, tens or hundreds of thousands of years, they still find living bacteria in the interstices between the ice crystals. So life is very robust in that regard. So are there fossils that you could extract from this? No one has succeeded in doing that, but that doesn't mean that they don't exist. What is the largest size of the void that you might find? I don't know the answer to that question, but it's certainly larger than clusters of bacteria.
Okay. Other questions. There is a request to stay in touch, and I see Bryce had waved a hand up, so go ahead and unmute yourself, Bryce.
Okay, it was a fascinating talk. Thank you. Lots of questions, and some more will come up if I think about it a bit. But does life require organisms?
That's a wonderful question. Thank you, Bryce. Depends what you define as life. Okay, there's been a debate in the community going back decades now about which came first, metabolism or genetics. And I'm of the belief, as I think most people in the field now are, that metabolism came before genetics. But what do we mean by metabolism? What we mean by metabolism is an autocatalytic set of molecules that take primary materials and a source of free energy and are able to reproduce themselves. You and I actually saw, and all the organisms that we know of fit this criteria. You and I can live on basic materials, basic sources of food, glucose, for example, and a couple essential vitamins and minerals. And we're able to synthesize the vast complex of molecules that make up our bodies. And this is actually true of most organisms. There are now plans to try to sample, well, that's not quite right. There are plans to send probes to some of the moons of the outer planets that are known to have subsurface seas. Titian, Enceladus, one of the moons of Saturn, and so on. We know they have subsurface seas because flybys of these moons have shown sprouts of water, geysers coming up through the crust. Enceladus, for example, is estimated to have a subsurface sea that is some 100 or so, I forget the exact number, kilometers thick. What happens if they sample this material that comes out on these geysers, and they find that it's loaded with complex organic molecules, too complex to have occurred by any random process, ones that require a history-dependent process? That's what life is. that exceeds the thermal lifetime of these molecules on geologic timescales. It could be that what you have is an autocatalytic set of molecules in the sea without any cells whatsoever. In which case you might ask, what is the living organism? And I would say it's the sea itself.
How about the lateral communication between two bodies of rock 10 feet apart or 1,000 miles apart?
Well, these organisms do move.
Through rock?
Yes. They're not moving through rock. There's a pore structure in rock. Even in deep rock, if that were not there, you'd never be able to get oil out of deep rock. And that's true also for ice crystals. Not commonly appreciated, but even if you have a solid mass of ice, the junctions between crystals consist of a film of water, which is liquid-like, and bacteria can navigate it. That's why you find organisms, microbes inside this ice that's drawn from a kilometer down in Antarctica. And as we now know, there are lakes that are heated up by heat coming up from the Earth underneath the ice, underneath 2 miles of ice in Antarctica. And these lakes, the largest lake, Vostok, has been penetrated by the Russians in a drill experiment, and it's loaded with organisms. It has not seen sunlight for a very, very long period of time. These organisms are found not only inside the water, but in the ice hundreds of meters above that water. And they're living. It's remarkable, but it's true.
Very cool. There is another question in the chat, and again a request to stay in touch. The question is, how coupled do you think the two biospheres are? What effect does what we do up here have on life down there?
Oh boy. Remember my comment about it's hard to sterilize the Earth? Okay. Let's suppose that tomorrow Mr. Putin and Mr. Trump become crazier than they are. They decide to set off every nuclear explosive in their arsenals. In fact, let's assume that tomorrow every nuclear bomb on the surface of the Earth goes off. You and I would be very, very uncomfortable. It would rank as one of the great extinctions of the Earth. These organisms would not even notice it. Okay, so what kind of communication is there between what we do and what they experience? Probably very little for most of them, certainly very little for a very, very long period of time for the deep ones. That's not to say that we can't affect them chemically over a very long period of time, but what I like to put it is we do not have the technology to sterilize the Earth. We don't have the technology to affect these organisms in a way that would kill them all. We don't have technology that's close to what happens when you put a 5,000 kilometer crater on the surface of the Earth. So they're pretty robust.
Yeah. Other questions? William, go ahead.
Is there any indication that there are viruses that attack these bacteria?
Oh, yes. A lot of viral material is brought up as well. I have spoken about that. In fact, a lot of that viral material, which I haven't included in my estimates because I do not consider viruses to be living. They are something which is their molecular component. It only comes alive when they're incorporated into an organism that has the metabolic machinery to be able to produce proteins. And yes, there are lots of viruses down there. We can see that DNA. We can see the viruses that attack a lot of these organisms. So the viral bacterial game of play goes back a very, very long way.
So RNA viruses as well?
I'm sorry?
RNA viruses as well as DNA viruses?
Yes.
Wow.
Yeah, it brings up the question of what you mean when you say that something is living. People have been arguing about this definition for a long time. Can I share screen again a moment, Don?
You're still sharing, actually.
Oh, okay. I'm sorry.
That's okay.
Okay, so here's a definition that I like. In fact, it's the one I came up with. I'm not unique in coming up with this. Many people have come up with similar ones. Freeman Dyson many years ago argued that there's little point to try to come up with a definition of life. No one has succeeded in doing it. What's important is to know what happened. I agree with the second point. What's important to know is what happened, but I disagree with the notion that coming up with a definition is a useless endeavor because it forces you to think hard. So a living thing, at least according to what I believe, is an autocatalytic chemical network, one that's able to create its own molecules. And you can refer to Stuart Kauffman to see that in more detail. That contains a high assembly index. That's to say these are molecules which by a quantitative measure, such as the one developed by Leroy Cronin and Sarah Walker, means that these molecules are just not randomly going to come together. They require a prehistory of autocatalysis, a slow buildup capability. Molecules that this chemical network sustains itself by absorbing material from and dissipating free energy of its environment. If that's true, death occurs when the machinery of autocatalysis becomes irreparably degraded. And I'd like to say there's a third state that we tend to ignore. Some living things may exist in a state of stasis, which for a long period of time, autocatalytic activity approaches zero. Those spores that were inside those brine inclusions, I think, would be a good example of this. Now, you notice this definition says nothing about cells, says nothing about DNA, says nothing about genetic material. And that's why I say that if we were to find molecules inside the waters of Enceladus, for example, that are of high assembly index and shouldn't be there over geologic time, they must be generated by an autocatalytic network, and why I believe that the entire ocean, in that case of Enceladus, should be thought of as a living thing. Not everybody is going to agree with this, of course.
Yeah, I just noted the echoes of the Horta on Star Trek.
Yes.
And Rob's got a hand up. Go ahead, Rob.
Yeah. So I just wondered if enough is known about the phylogenetics of this diversity of things that are deep beneath the Earth's surface to know if some of the earliest I guess this is the right way to say it, the earliest representatives or descendants of the earliest representatives of early bacteria and archaea are found in these places, and whether most of what we're finding deep beneath the surface are things like you suggested for the Carboniferous bacteria in fossil coal, whether it's mostly relatives of things from the surface that have adapted to life deep beneath the surface, or whether it's mostly things that have been down there a long time and have and are, effectively independent of modern lineages.
I would argue, but I think that the straightforward answer to your question is no one really knows. Okay, partly because no one really knows, other than from these fossil impressions, what some of the earliest microorganisms look like. But I see no reason to assume that the earliest cells had to have evolved on the surface. And as I pointed out, the argument about the late heavy bombardment really suggests that they would have had a very hard time. So I think the straightforward answer to your question is no one knows. I'm not a microbiologist. I'm certainly not a microbiologist that deals with deep organisms. Perhaps a microbiologist might give you a better answer to the question. I'm a structural biologist, yeah. Structural physicist. But I don't think anyone really knows. That's okay. One of the wonderful things. One of the wonderful things about working in this field, and it's something that I love to tell students. Students very often come into biological physics and they say, are there any good problems left to work on? And I just Wait a minute. Okay. We have to have some humility. We really, and this lecture, I hope, has been an example of that, understand very little about what we don't know. We don't know what we don't know, but there's certainly a lot to be known. And we're discovering more all the time. So the great thing about working this field is that there are so many questions to be answered. And once you think of them, you can start dreaming up ways of answering them.
Yes, indeed.
Thank you. That's a very intriguing answer.
Yeah, there are a couple of comments in the chat. Liz says, stunned, read the odd article aimed at those outside the field and was not aware how much research has occurred and the astonishing results of that research. And George notes that Carl Zimmer makes a serious effort to define life in his book, Life's Edge. Other questions?
I see William Bryce raising a hand there.
Oh, go ahead. Go ahead, William.
Yes, I was just wondering, given that this stuff seems to exist below the surface, would we find the same thing on Venus and Mars?
I don't know.
Maybe we're looking in the wrong place.
Well, certainly NASA has to justify its existence. So certainly they are looking on Mars. I mean, there's a substantial effort, as you well know, to try to bring back samples from Mars. No one has done that yet. We do have meteorites that are known to have originated on Mars. There are various ways to know that. And there was a big dispute some years ago, a decade or two ago, about squiggles in that rock where someone was claiming that they were bacterial organisms from these Martian naturally falling rocks. But we don't know. Okay. And of course, no one has brought anything back from Venus. The surface of Venus is, really hot, 800 degrees centigrade, or something like that, because it's got a greenhouse environment that is way up there. So there's just no answer to that because no one has samples. Is it plausible that there was life on Mars? I think it's perfectly plausible. Is it plausible that life might still exist in microbial form on Mars? I don't know, but certainly a lot of people would like to find out. And there are plans to actually go there, get enough samples, and try to answer that. What the rover missions have shown is that there are lots of sedimentary rocks. There was a lot of water at one point in Mars history. We don't know exactly when that time terminated, but chances are it was considerably after 500 million years. If life existed on Earth by the first 500 million years, could it have existed on Mars during that same time period? Well, it seems plausible. Mm-hmm. And if so, can we find traces of it? And for that matter, could it continue to exist inside places where we now know that there's subsurface water, such as on the poles of Mars? So just juicy questions. We don't know the answers to them, but there's sure a lot to think about and a lot to be done.
And a thank you from Paul, who had to sign off. And another question from Noreen. Have studies been done on the mantle rocks at the surface of Newfoundland?
On the mantle rocks, yes. Well, it depends what you mean by mantle rocks. You mean by rocks that have been brought up that we know, partly because of serpentization, that came from the mantle.
Yeah, I think that's right. The way I read that.
Right. Studies have been done. Have people found evidence of microorganisms in those rocks? Not to my knowledge. I remind you that trying to find evidence of microorganisms in rocks that date back to the time periods of when they were brought up is hard in the first place. Even if they were never cycled through that kind of deep metamorphic process. So I don't know of any, but I haven't searched the literature to be absolutely certain of that answer.
Other questions, comments? I will ask one. How did you end up getting interested in this sort of thing?
first of all, I have to say that this is not the primary thing I do for a living.
Mhm.
At this point, I'm emeritus, but
Yep.
prior to that, in addition to teaching, most of my work has been based on trying to study block copolymers, trying to build technology really to do X-ray kind of studies on molecules. My group has been heavily involved in developing, for example, the detectors that have been used to collect a good fraction of the structural biological data from proteins that exists. And from that point of view, I was on an NIH study section in I guess it was 1985 or 1986. And there was a proposal by a group led by Fred Richards at Yale, where they wanted to do the structural biology, determine the molecular structure of hen egg white lysozyme at 1,000 atmospheres. Why? Well, they just thought it'd be interesting to do. Okay, and I was very intrigued by that. And they did do that structure. It was the first high pressure protein structure ever done. And then I couldn't help but notice that they came up with answers that I found very hard to accept. It turns out the answers they came up with, namely that the structure doesn't change much, and that it's very hard to do these kinds of experiments, both were right and wrong. It turns out that there were small scale changes inside the structure of egg white lysozyme. And I determined at this point that I'd like to show that the small scale changes are really important. That's elaborated in that crystallographic lecture that's on the DAISY website of how we came to that conclusion. And the other thing I noticed is that because they said that this stuff is really hard science to do, the crystals tended to crack when they went to 1 kilobar. We found that when we started pressurizing protein crystals, they're actually extremely robust. Turned out to be a very strange coincidence that they were using salt conditions, which caused the crystals to crack. So we then started doing structural biology of proteins and analyzed several dozen. So I started by being unhappy about the one study that had been done on protein structure determination at 1 kilobar, and determined that I do structural biology. Let me look at what happens when I look at proteins, an array of proteins at high pressure. So we got involved in that way.
Well, thank you.
I have to point out one more thing, Don. You might
Yep.
ask, who funded that?
Who funded that?
Okay. I couldn't get anybody to fund it. So I had to do a cheat. The Department of Energy funded me to build the X-ray technology to do protein structure determination, these detectors for that. And that's what led me to Cornell, why I was director of the synchrotron facility for 17 years. And what I would argue to my DOE funders is, look, I'm in a university and I train students, and students do not get a PhD for making a widget. They get a PhD for making a widget and doing some science with it. So along with building detectors, I'm going to have each of the students use these detectors to answer some scientific question. And DOE accepted that answer. It said, as long as you're making detectors, we don't care about that, but that's okay. So in effect, I got the agencies to agree to let us do these kinds of science, these kinds of experiments, simply because there was a product in the process that they wanted, which is the technology they could use at the synchrotrons, which in the United States are primarily, exception of one at CHEST, run by DOE.
Okay, we've got one more question in the chat, and then I'll tell a very brief story about the structure of scientific revolutions. And so the one more question is, wouldn't you think that membranes would be a big field to study?
Oh, absolutely. That's actually where I started. So if you go into the third of those 3 lectures, it finds entirely devoted to the effect of pressure on membranes. And it turns out that membranes are the most pressure sensitive part of cells, because the compressibility of lipids is very high compared to the compressibility of proteins. Aqueous proteins have a compressibility of about 5 parts per billion per atmosphere. That's the same compressibility as aluminum metal. They're really compact and tight. Water is about 50 parts per billion. Lipids are about 100 parts per billion. So if you're going to see an effect, it's going to be in the membranes primarily, because that's where the structural changes are.
Very good. Okay, and I will put a link in for donating again to PRI, and just very briefly say that when I had freshly moved to Hamilton, New York, to Colgate University, when my older daughter was about 3, we were living out of boxes, and she wanted me to read to her, and she pulled a book out of the box and brought it over to me and said, read, Daddy. And so I said, okay, The Structure of Scientific Revolutions by Thomas Kuhn. And she said, Thomas Kuhn is funny. And he certainly is in certain ways, but memory will stick with me for the rest of my life. She's now 25.
How old was she at that point?
She was 3.
Oh, so what did you read to her? It's not exactly a book for a 3-year-old.
I started reading the introduction, and she was with me for maybe a paragraph or 2. But
Okay.
I did not read the whole book to her. All right. Well, thank you. That was a very, very great session, and I really enjoyed it. And we are off in 2 weeks for Thanksgiving, and back one more time in December for our 100th session with a panel of Science in the Virtual Pub alumni talking about how their science has changed and how it might change in the future. I'm very much looking forward to that discussion, so I hope you'll all join us. And
excuse me.
Yes.
Excuse me, sorry.
Yes, go ahead.
I want to ask to Dr. Gruner if I keep in touch by email, please.
Yes, my email address is in the first slide. It's smg26@cornell.edu, and be happy to communicate.
Put that in there and make sure I spell it right in the chat.
Well, yes, please.
All right. All right. Well, thank you and good night.
Okay, thank you.
In a month.
How can I preserve the chat? Because I would like to see.
You should be able to click on the 3 dots at the top, I think. Yes. The 3 dots up at the top of the chat, and the first thing there is Save Chat.
Where does it save it?
It saves it on your computer.
Okay, very good.
Yep.
Thank you.
Thanks, Doctor.